This sodium hydroxide test. In this we are going to confirm ammonia liberates from the protein. Okay, and this test is called as sodium hydroxide test. And in this we have taken three different samples, sample A, B, C into test tube 1, test tube 2 and test tube 3, 1 ml. Then I have added 40% sodium hydroxide 1 ml in each solution. Then what is 40% sodium hydroxide? 40% sodium hydroxide is 40 grams of sodium hydroxide dissolved in 100 ml of distilled water. Okay, and I have made a solution and I have added to this. And once I have added, shake the contents. There is no change in color. No change in the color. But the only thing is, when you dip this with a litmus paper, this is the litmus paper, and when I dip it into this, it will change to blue color if a protein is present. If there is no protein, there is no change in the color. Okay, we will see whether the protein is present or not and ammonia whether it is liberated or not. There is no change in the litmus paper. Okay, now we will see the second sample, sample B. There is no change in the color. Now if I take it the third sample, just observe it like this. There is a change in the color of the litmus paper to blue color. That means ammonia has been released from this sample, from the protein. Okay. Now this is one method of liberation of the ammonia testing from the protein by litmus paper. Now we will go for the second process, that is take one glass rod and dip it in HCl solution. When you put it near the sample A, there is no fumes formation. Okay. Similarly, if I put second solution, there is no fumes. No fume is formed. Light fumes are formed. But if I put it in the protein sample, you can see white fume is formed. Can you see? दिख रहा है धुआं? I'll put it again. White fumes are formed. Okay, as you can see here, white fumes, and this white fumes indicates the presence of ammonia liberating from the protein. Got it? That is the end of the second test. First test is biuret test, where you get purple color with the sample, and in second test, sodium hydroxide test, fumes are formed. These are the two qualitative analysis tests for the proteins. Okay, thank you. We will go for the next test in the next class.